Okay, students, after having some basic information about techniques that could be used to determine chromatin structure and function, let us discuss little bit detail of chromatin immunoprecipitation assay and further downstream reactions which could be possible using this technique. By the way, there is a separate lecture on CHIP present on my channel. If you haven't seen that video, please pause this video and go back and watch that video first, and then come back again to better understand this overall process. Uh, okay. Uh, Chromatin immunoprecipitation or CHIP has two different categories. One is called native CHIP or NCHIP, and another one is called the cross-linked CHIP or XCHIP. Native CHIP mean you wanted to target the proteins which are abundantly present with chromatin, and because they have natural affinity to bind with the chromatin and DNA, so that is why you do not want to disturb this interaction using different chemicals. So that is why it's more or less a native environment when you are performing your CHIP in this native environment. That is why you wanted to do you are preferring this and native CHIP, because by employing different type of chemical and reagents you could somehow slightly modulate the nature of different types of interaction which are going on in chromatin. So that is why this type of technique, this type of CHIP technique is used when you are interested more or less in native interaction information. So when this there are advantages and disadvantages of this technique. For example, this technique is suitable to analyze histone marks and other abundant target protein. Moreover, this do not require cell fixation. That is, it's more or less more natural environment, so you do not have to fix the environment of the cell, so you are not going to dehydrate it. Secondly, uh, being more accessible chromatin procedure, it facilitates formation of antigen antibody complex because of their natural affinity it will be formed. However, this process require long duration to perform. I mean, because it's a natural environment and it requires longer duration for complex formation to be formed, and that is why you may have to put it for overnight. uh, more Moreover, when the complex has been made, you have to shear that shear that chromatin using enzymes only, because you cannot go for mechanical uh, lysis. This will disrupt this native interactions which you wanted to preserve through this technique. Secondly, less abundant transcription factors or proteins, for example, uh, maybe some other modifying enzymes cannot be captured through this technique. So that is why this has a limitation. The other technique is called the cross-linked or XCHIP technique, which is suitable for proteins that are not directly bound to the chromatin, but they are associated proteins in the chromatin. For example, some transcription factors and other modifying enzymes, you could focus them. You could be interested to get them. uh, you could be interested to know the nature of their interaction with the chromatin. The advantages of the technique are there. It normally requires small amount of time. For example, it is not that a longer process as compared to the native chips. So you might require only 5 hours. Chromatin shearing is possible through sonication or mechanical stuff. Unlike the NCHIP. Uh, Similarly, it can investigate wide range of chromatin associated protein, even they are present in small quantities. So, for example, transcription factors and other modifying enzymes could be targeted through this. But, however, uh, this had disadvantage because you need to cross-link the chromatin, and fixation is required. Cross-linking means you wanted to uh, fix the interaction in real time at that particular time point in which you have performed that experiment. So because you wanted to document each, even a slight interaction which is possible with chromatin and any protein associated with that, so you don't want to lose it during the process of chromatin formation, during the process of immunoprecipitation. So that is why you have to fix that. And mostly normally the fixation is done using some chemicals. For example, normally the fixation involves the hydration process thing. UV cross-linker could be used, formaldehyde could be used, at any other chemical method could be used for fixation process. It do not work. It does not work chromatin in native state, so that means could be possible the native proteins which are present in the chromatin uh, do not show that similar result as you perform a native chromatin immunoprecipitation technique. Moreover, cross-linked chromatin can occasionally mask epitopes of some antibodies, affecting their natural affinity to bind with the chromatin. So this overall process, chromatin immunoprecipitation process, is shown in this video. So now this is the genome. For example, this is the chromatin, and this is your protein of interest, which is called POI, which is bound to the DNA. Now, in other words, this is a nucleosome in which your protein of interest is present. So what will you do? You will either go for native chip or XCHIP. In both cases, you are going to cross-link them, and once you have cross-linked them and you added the antibody in this mixture, this antibody will bound to this protein of interest, and by the end of the day, your protein of interest will be separated, and now you can digest it, you can purify it. So you will get two fractions. One fraction will contain the DNA, this DNA, and other fraction will contain the protein. So you have two different things, one is protein, another one is the DNA. So you could use this DNA for going chip on chip, you could go for CHIP-seq and any other thing. On the other hand, you could use this protein जो कि आपने अभी purify की है, this protein for SES page analysis, mass spectrometric analysis. Maybe you wanted to do hybridization and any other thing which you wanted to do, you could go further. So this is a simple workflow of chip on chip analysis in which you perform the chip. This is the normal chip, and by the end you get the DNA and you go for chip analysis, which I have already discussed. That you might be interested to go for CHIP-seq, in which in that case you will separate the DNA from this chromatin using chromatin immunoprecipitation technique, and by the end of this you will have your DNA, and you do not know any gene of interest in which you are interested. So you wanted to go for genome wide, you go for Illumina sequencing. It's a genome wide approach in which small adapters will bind and will amplify the whole genome. So it will give you read, and you will know by the end of the day what type of genes were present. So okay, this is the picture which tells you these adapter proteins and how they are amplifying, and you will get the fluorescence by the end of the day, and you know everything. So this is the CHIP-seq. So you, by assembly, you could get something like this. So these peaks, so this is the chromosome, this is the normal reads, and you will get the peaks. And these peaks, when localized, will tell you this peak belongs to this particular gene of interest, or this peak belongs to this gene of interest. And you know that, you know through the tag density what type of genes were enriched and what type of genes were depleted in your CHIP-seq analysis. So you see two terms, enriched and depleted. Enriched for those which are abundantly present, and depleted those which are not present so abundantly, are very
सर जी
जी जी
एडाप्टर प्रोटीन का आपने बता दिया कि ये डीएनए के साथ बाइंड करता है, ये
अडेप्टर प्रोटीन
आर्टिफिशियली
जी जी अडेप्टर्स जो है ना, वो स्मॉल डीएनए टैग्स हैं जो कि रीड करते हैं। जी आप क्या कह रहे हैं?
सर ये
मैं
आर्टिफिशियली प्रिपेयर करते हैं या ये टैग जिनोम में मौजूद होता है, इसका
एक्चुअली ये
सीक्वेंस
तो
लेके हम डिजाइन कर
ये तो जस्ट नॉर्मल रैंडम हेक्सामर्स नहीं होते। इट्स समथिंग लाइक दिस। सो दिस इज, these are meant to, these are meant to amplify anything which is present. So they are not, these are synthetically made and they are present. So I mean, what is the probability of getting AAAGGG? So this is normal, just a normal tag. You could use that and it will amplify everything which is present in your genome of interest. So these are normal random adapters. It has nothing to do with any specific thing. ठीक है? So इसका मतलब ये है कि आप जब आप जीनोम वाइड कर रहे हैं ना, तो आप, यू कैन नॉट बी स्पेसिफिक, आपको खास सीक्वेंस अरेंजमेंट दे नहीं सकते। अदरवाइज तो वो सिर्फ बहुत सी बायसनेस क्रिएट करेगा। वो बायसनेस इस तरह की होगी कि कुछ सीक्वेंस प्रेफर हो जाएंगे, कुछ सीक्वेंस प्रेफरेबल नहीं होंगे। ठीक है?
Yeah.
So the adapter protein बाइंड करती है और उसके बाद चल जाती है। ओके, देयर इज अ कंपैरिजन ऑफ डाटा जो कि दो डिफरेंट किस्म की टेक्निक्स के साथ है। क्विकली, क्योंकि हम आगे की तरफ जाएंगे। दीज आर चिप-seq वर्सेस चिप ऑन चिप डाटा। दीज आर टू डिफरेंट टेक्निक के थ्रू एक ही डाटा प्रोड्यूस किया गया। लेट मी गिव यू अ क्विक ओवरव्यू ऑफ दिस स्लाइड। सो दिस द अपर पैनल uh, tells you about the experiment which was done through this chip chromatin immunoprecipitation. So this is chip employed for H3K4 trimethylation. So that means lysine 4 methylation on histone H3. So this antibody was used, an antibody which could identify lysine 4 trimethylation at histone 4. So that is why it is called H3K4 trimethylation. So an antibody which could identify this was used to go for chip technique. So this is a native chip experiment where the natively generated antibody antigen was used to highlight this overall thing. So by the end of the chip analysis, uh, by the end of the chip analysis, uh, the reads were made. The experiment the data was produced using two different techniques. One was seq technique, another was one, and other one was another chip. So this is CHIP-seq and chip on chip. So you could see both of these panel. Both of these panel use DNA generated through this chip which has H3K4 trimethylation antibody. I am very clear on that, और मैंने आपको बड़ी सराहत के साथ ये बात बताई. And by the end you have the DNA, and you analyze the DNA using Illumina sequencing, and you analyze the DNA using chip on chip. So both of these, one thing you could see here, both of these experiments give you somehow the same pattern of genes, just because this is a genome wide thing. So this is 20 kilobases of assembly जो कि read की गई CHIP-seq के जरिए और chip on chip के जरिए. You could see that the peaks are present almost at the same location. जहाँ पर CHIP-seq ने दी, वहीं पर आपको chip on chip का डाटा भी नजर आता है. But one thing which you need to be uh, but there is a difference. आप identify कर सकते हैं कोई difference इनमें से? Can somebody tell me what is the difference?
ये
सर चिप
चिप
एंड चिप
सीक्वेंस
में
और
थोड़ी सी ज्यादा मतलब कि आई हैं
नीचे
पीक्स,
दो जो
चिप
रेड
सीक्वेंस में थोड़ा सा
है
कम है ऐसे लग रहा है।
अच्छा, वन बाय वन
स्पेसिफिक
अरफान बात कर रहे हैं। अच्छा ठीक है। और आप कह रहे हैं कि चिप ऑन चिप में पीक्स ज्यादा है।
ज्यादा स्पेसिफिक नजर आ रहे हैं। चिप सीक्वेंस में थोड़ा सा कहीं वो क्या कह रहे हैं कि कम हो रही हैं चीजें, थोड़ा सा वो लीनियर सी जा रही हैं,
आप
बहुत ज्यादा
आपको लगता है चिप ऑन चिप स्पेसिफिक है और चिप सीक स्पेसिफिक नहीं है?
थोड़ा सा स्पेसिफिक नहीं है। मीन्स कि सर जहाँ पे बहुत ज्यादा पीक है तो वो दे रहा है, जहाँ पे छोटी माइनर पीक्स हैं उनको वो एक ही तरह का रख रहा है, जबकि चिप एंड चिप जो माइनर पीक्स हैं ना उनको भी डिफरेंशिएट कर रहा है।
Should it not be the other way around? For example,
सर चिप सिक्स जो है वो ज्यादा स्पेसिफिक है, जबकि चिप चिप स्पेसिफिक नहीं लगती इसमें, उसमें एक्स्ट्रा पीक भी आ रही हैं।
ओके, सो दिस विल बी द अदर वे अराउंड टू सी। आपकी बात बिल्कुल मैं एंडोर्स भी करता हूँ। दोनों में वन थिंग, चिप ऑन चिप के अंदर बहुत सी आपको पीक्स नजर आ रही है, चिप सीक के अंदर स्पेसिफिक पीक्स नजर आ रही है, जिसका मतलब है कि चिप सीक इज मच बेटर। इन अदर वर्ड, चिप ऑन चिप इज हाईलाइटिंग सम नॉइज, सो दिस इज द बैकग्राउंड। सो इसका मतलब है कि जिसके अंदर जितनी इट इज मोर सेंसिटिव, चिप ऑन चिप इज more sensitive but is not that specific. CHIP-seq is more specific but is not that sensitive. You see, there are two different terms. अब मेरी बात समझिएगा जरा। Sensitivity and specificity, they are more or less inversely proportional to each other. ठीक है? Specificity and sensitivity. आप प्लीज अपने माइक ऑफ कीजिए अगर आप कुछ कहना चाह रहे हैं तो। ठीक है? Chip on chip is more sensitive than CHIP-seq. This tells you the reliability on the data, how reliable a data could be. For example, I would prefer more CHIP-seq data because this will highlight some specific peaks. So similar thing is down, you आपको यहाँ पर trimethylation lysine 27 की histone H3 के ऊपर भी नजर आ रही है। This chip was done using this, which could target uh, trimethylation status of lysine 27 on histone H3, and the again the DNA was processed by two different techniques, CHIP-seq and chip-on-chip. Still you could see that more or less the similar pattern is present on both the techniques, both the data assemblies. But one thing again, the chip-on-chip has some unrelated, chip-on-chip has background, or it is more sensitive because it gives you lots of peaks, which and those peaks are not present, are not that prominent in CHIP-seq data. That means CHIP-seq is more क्या कहते हैं इसको, more specific. So I hope with this understanding you are able to understand how you could go, how you could plan your analysis, you could plan your research if you wanted to do something in chromatin research. So you have the idea now that you could target chromatin, and you could target chromatin using its native protein, and you could target chromatin any other protein. So it's not always that कि आपकी आप native के साथ ही काम कर रहे हैं, sometime आप transcription factors के साथ काम कर रहे होते हैं, या फॉर एग्जांपल कोई मॉडिफाइंग एंजाइम हो सकता है, जैसे हिस्टोन एसिटाइल ट्रांसफेरेजेस है, सो यू कुड गो फॉर XCHIP, and by the end of the day you could process the data in a different way. I have an assignment for you people, and that assignment is about chromatin conformation capture. I haven't give you much detail about that because I wanted you people to search that technique and make an presentation. I would make schedule of presentations for you people after midterm exams.